Hello and welcome dear learners to the NPTEL course on Nutritional Epidemiology, a way towards a healthy life. In the module 2, the study designs and epidemiological methods for the chapter 19, we'll be studying about the biomarkers in nutritional epidemiology. So what are the biomarkers, types of biomarkers in nutrition, characteristics of a good biomarker across study designs and biomarkers in validation studies, its strengths and limitation and the challenges in nutritional epidemiology will be studied over here. So in the nutritional epidemiology, a biomarker is an objective, a measurable biological indicator of dietary exposure, nutritional status or a diet related disease pathway. So if you take this, it is unlike a subjective self-reported dietary assessment tools like a 24-hour recalls or food records. So that are prone to recall bias and measurement errors. Biomarker capture the physiological reality of what is absorbed and metabolized. So where you find nowadays there is a great importance of biomarkers which is being increasing, and the biomarker examples like urinary nitrogen to assess the dietary protein intake, which is very important in the kidney patients. Serum ferritin to assess the iron stores actually, which is very important for iron absorption studies. Plasma ascorbic acid, vitamin C to reflect the vitamin C intake is very important. Urinary sodium, the salt intake, which is very important to understand for the cases of cardiovascular disease and particularly hypertension. HbA1c, long-term glucose metabolism for the patients of diabetes and even glucose index. What are the type of biomarkers in nutrition? The biomarkers of exposure intake, biomarkers of nutrition status, biomarkers of effect and biomarkers of susceptibility. So biomarkers of exposure intake like used to estimate the absolute or relative intake of specific nutrients or dietary compounds. So there might be subcategories to be stated. Recovery biomarkers, it represents a known constant fraction of ingested nutrient recovered in excreta over a specific time frame. For example, 24-hour urinary nitrogen, a protein intake, doubly labeled water energy expenditure. The other category is concentration biomarkers, which is correlated with intake but are heavily influenced by individual metabolism, absorption and hemostatic regulation. Cannot provide absolute intake figure but useful for ranking individuals. Example, the serum folate, plasma carotenoids. Then comes predictive biomarkers, which exhibits a dose response relationship with intake but not have the complete time bound recovery of markers. Example, a 24 hours urinary sucrose or fructose, the dietary sugar intake. So it can be seen that the biomarkers of exposure estimates the absolute or relative intake of specific nutrients or dietary compounds. On the second hand, the biomarkers of nutritional status. So these biomarkers reflect the body's nutrient reserves of functional capacity, representing the cumulative effects of long-term intake, absorption and utilization. If you talk about the examples of RBC folate, can be taken as what it represents. It is a long-term folate status which reflects the body stores over weekends to the months. Okay, then is the serum folate. Serum folate, recent folate intake reflects the short-term status. It is very important to see a neural tube defects in children's, particularly which is as being status as depletion of folate levels in a mother in pregnancy. Serum ferritin, iron restores in the body. This is a biomarker which is give you the nutritional status of serum ferritin. Serum 25, OHD, the vitamin D status which is being pointed over here. So this indicates the adequacy and it comes as a deficiency or excess of a nutrient in the body. Okay. The third biomarker's effect. This indicates an early biological response or physiological alteration resulting from a nutritional exposure. This biomarker acts as an intermediary step before a clinical disease manifestation. For example, HbA1c reflects a long-term effect of chronic carbohydrate load, insulin resistance. Second example, DNA adducts indicate oxidative stress and damage to DNA. And the LDL cholesterol early effects of dietary saturated fats on lipid metabolism. CPC, where the CRPC markers of systematic inflammation which is occurring there in the body, which is an indication of some sort of inflammation which is taken care from longer period. So these are useful for understanding the mechanism and predicting the risk if you are not understanding to take ahead and interventions related to this. A biomarkers of susceptibility is the fourth type, which is an intrinsic factors often genetic that influence how an individual metabolizes or responds to a nutrient. It serves as an effect modifiers in the diet-disease relationship. Examples are MTHFR gene mutation. It affects the folate metabolism, and hypohomocysteine regulation influences individual requirements and response to folate intake. Lactase gene variants affects lactose digestion, lactose intolerance. APOE gene variants influence lipid metabolism and response to dietary, that is fat and cholesterol. GST polymorphisms affects the antioxidant effects and response to dietary compounds. So this glutathione S transferase polymorphisms, and these examples states that the biomarkers of susceptibility has an importance for precise nutrition and personalized dietary advice, which is coming as one very important part of this. What are the characteristics of a good biomarker? Biological and clinical relevance validity. If you talk about the biological plausibility, the molecule connecting to an organ like heart or lung with mechanistic pathway diagram, and a high sensitivity as a target with many points clustered very closely together and an early alarm. High specificity, a filter on a target where all points are only within the bullseye. Responsiveness, a slider or a thermometer, and changing level linked with a changing exposure. Also, it is an added predictive value to show that a graph showing a dotted line into future outcomes. So you can understand there is a great biological and clinical relevance which is proving as a validity from this good biomarker. Okay, there is an analytical performer which is being giving a reliability which can state that if a biomarker has to be good, it should come up with accuracy and precision, followed by reproductivity and clear reference ranges and robustness. Very important that ideal biomarkers are balancing these features of political biomarkers, trait in cases channels, fragment to thermodynamics in the features of PUs as in good biomarkers. So if you talk about the practicability and feasibility, it is minimally invasive, it should have a sample stability and cost effectiveness followed by high throughput, as if an automated lab robot arm moving multiple tests of samples, which can say that it can be seen as a high throughput biomarker and stated as an characteristics of that biomarker to be coming as an acceptance. Biomarkers across different studies. How do we see biomarkers in different studies? In cross-sectional, case-controlled, prospective cohort and RCTs. So in cross-sectional, the snapshot, if I have to take it, it is like the time simultaneous measurement where exposure as an outcome, exposure as in being seen as a biomarker to come up with the outcome, prevalence assessment, generating hypothesis, cost effectiveness. So it is something which is giving a reverse causality which came first, and this can be seen to state that a biomarker is being proven to be very important in the cross-section study. In the case control study, it is being seen something which is studying the rare diseases and there is no recall bias which is being proven to state that it is good for long latency diseases. The disease effect can change markers level. So present versus past. In the prospective cohort, looking forward where you are trying to take from baseline to future, from a cohort to a healthy cohort, and you are understanding with the years, it is a measurable baseline in healthy cohort and predict the future risk. It establishes the temporality and it avoids the disease effect. RCTs, randomized control trial, the experiment where you say intervention versus control. These biomarkers are being seen as a pre-intervention and a post-intervention. So experimental design, proving causality, surrogate endpoints and monitoring compliance. So biomarkers across study designs are being seen representing their own benefits. Biomarkers in validation studies. If you talk about the correlation, actually how it is being seen, the core rationale to see the independent measurement error, wherein the traditional validation and a biomarker validation, where you can see the correlated efforts which comes as to understand the correlated errors as from junk food underreporting bias to inflated validity, and an independent error where a cognitive versus lab metabolistic thing actually where a variation is there. Application of specific biomarkers, recovery biomarkers, concentration biomarkers and predictive biomarkers have their own application which has been seen over a period of time how the comparison, the rare individuals and validation are being seen as sometimes as a very important way actually to put these biomarkers on the place of validation studies. The methods of triads where test and reference method are being seen as an estimate validity coefficient versus a biomarkers which is giving you a structure equating model where SEM are truly being taken as an estimate for identifying the biomarkers. From validation to calibration, if you understand the entire cohort versus to apply it in calibration for the main cohort, you may see the application of this as to the main cohort. And there is a prediction actually which comes to state that how this validation is proving the benefits for this part. Key limitations and challenges may also be backed by stating that there is a lack of comprehensive markers, logistical burden and temporal mismatch which can give you some set of backs for stating that the biomarkers validation also come up with some sort of limitations. If we talk about the strength and limitations, the strength comes with objective measurement and it bypasses the memory and biases where you may talk about the years back or even for the future or even sometimes the biases actually which comes over that the biomarkers prove to be little benefiting. Also capture the internal dose. So it is very important that it accounts for bioavailability, the digestion and bloodstream, absorption, utilization, persons and it is comparison also it is very benefiting for this part. Also on the separate distinct where you talk about FFQs and separate distinct, it is being proven to state as a benefit for this part. You can have biomarkers like HbA1c where there is a direct link to the disease mechanism where here in this case it is with the heart and early cellular changes which may say that it is a benefit where you can establish the direct link to the disease mechanism. So on the strength part you may find that there are objective measurement, capture internal dose, independent measurement error and direct link to disease mechanism. Where versus on the limitation as already stated previously, temporal mismatch where the short term snapshot, habitual diet, biomarkers. So you may find there is a far distinct actually and that can come as a limitation. Hemostatic regulation. So you may say here it is a nutritional intake and there is a blood level. So there is sometimes a mismatch actually which may say that bone stores, tissue buffering. So pulls from tissue to maintain the status quo. That can be seen as a body regulating the levels also. High cost and logistical burden where from the very start as you can see that biological markers actually are being seen as for collection, transport, analysis. So it takes more like a prohibitive expense actually, and you can say there is a very logistical burden actually to take this as a major limitation. Invasive nature and burden. So you may find that it is very painful, inconvenient, and sometimes for time. So this is being seen as also sometimes which is taking more time actually to state it, and also to be counted as a major limitation. Lack of comprehensive markers actually. So no markers for total fiber, no markers for complex diet, no markers for different patterns. So and others actually you can say, so comprehensiveness is not being covered in biomarkers, which is being seen as a limitation. So if you talk about the limitations on a part, you can take temporal mismatch, hemostatic regulation, high cost and logistical burden, invasive nature and burden, and lack of comprehensive markers actually not being seen as. So in the challenges in nutritional epidemiology, we can talk about reductionism versus holism. So protein molecules versus dynamic interactions, vitamin molecules, and single nutrient. So diet is complex and synergistic. Biomarkers can miss this interaction, and this can be seen as a major challenge. Okay, where we have more a flow or a process towards holism. Okay, where it is giving a reductionism towards understanding a particular molecule. Lack of biomarkers, particularly for fiber, as being stated in the previous section, whereas polyphenols, dietary patterns. So many critical components cannot be measured directly with the biomarkers. Inter-individual variability, where male versus female, organs, and so many things like the genetic, age, metabolism cause vast differences in biomarker levels are being seen from an individual single meal to a complex. Confounding and measurement errors are no doubt invariably where you may say inflammation, disease, smoking, and lab variations skew the results. Ethical and logistical constraints, 24-hour yielding connection, cold chain, logistic, participants, high cost, where you may find the samples are invasive, difficult to collect, transport, and analyze, which makes ethical and logistical constraint as one of the important challenge. Need for multi-omics, you may say metabolomics, proteomics, transcriptomics, and epigenomics. So integrated with biological information is very essential to prove and reduce this. And lastly, ethics and equity, which is very important part in public health, where you can say the ethical consent and social determinants, which cannot be ignored, to state that to balance the equity part, the social connects the strict consent, ethical consent, cultural humility, and social context must prevent the stigma and bias. To overcome this, it can be stated that ethics and equity stands along with all other challenges in the nutritional epidemiology. In the summary to the biomarkers in nutritional epidemiology, which is stating the importance, understanding, and the usability of the biomarkers in nutritional epidemiology, it is being seen as an objective, measurable indicators, and it is being used to assess the dietary intake, nutritional status, and biological response to the diet. No doubt it has been stated to have a great importance in the current times in organizational status, actually to understand, especially in healthcare organizations, hospitals. But on the other part, it has an limitations, and they help also to overcome the limitation of self-reported dietary data. So it has one way actually a benefit versus to our limitations. By providing more accurate and unbiased measurement can be seen as something which is more towards an individual person. Biomarkers can be classified into exposure, status, effect, and sustainability types. Each of them have its distinctive role in understanding the diet-health relationship, where you can say they are widely used in observational studies, clinical trials, and validation studies, which improve the causal inference. As I stated, Causal inference is something which is being stated and to be used in association with biomarkers. So we may find the benefits of biomarkers in observational studies, clinical trials and validation studies. Despite the strength, the challenges and the limitations we may see as cost, variability and limited availability, which is persistent and can be seen something which is being putting them little out actually for taking as a choice. So overall, biomarkers are essential tool for advancing the precision of nutrition and strengthening the evidence in nutrition research. Costing, variability and limited availability may be seen as a challenge, but to understand the precision of nutrition and strengthening the evidence, it is very important that biomarkers are being coming up as an essentiality. So with this, we come in into this chapter, and taking these references ahead to understand more, you can see that biomarkers in nutritional epidemiology are being stated to be seen as very important. Thank you very much.